In this video, we'll look at how transcription is regulated. Eukaryotic promoters have 3 conserved sequences. Each eukaryotic promoter has 2 of these 3 sequences. The most common sequence is the Tata box. All of these promoters are bound by the Tata binding protein or TBP. Together, these sequences comprise sections of DNA involved in controlling the activity of genes and are hence called regulatory sequences. When proteins that regulate transcription bind these sequences, gene activity changes. Some of these regulatory sequences are shared between eukaryotes and prokaryotes, whereas others are very different. In yeast, an example of a regulatory sequence is the one that controls the transcription of 5 genes that are involved in the utilization of the sugar galactose. The regulatory protein that binds these regulatory sequences was found by scientists who named it GAL4. This protein binds a short stretch of DNA located just upstream from the promoter for all of the 5 genes required for galactose metabolism. Genes whose transcription is co-regulated are not necessarily clustered together. They share a regulatory DNA sequence that is near the promoter and binds the same regulatory protein. Such sequences are termed promoter proximal elements because they are closer to the promoter. Promoter proximal elements are located just upstream of the promoter and the transcription start site. They have sequences that are unique to specific genes, and they provide a mechanism for eukaryotic cells to exert precise control over transcription. As we can see in this image, transcription starts at the +1 site, also known as the start site, in this direction. The promoter is usually upstream of the start site. In addition, regulatory sequences, which in this case are promoter proximal elements, are present upstream of the promoter. Regulatory proteins can bind these promoter proximal elements, leading to the recruitment of transcription machinery and facilitating the initiation of transcription. There are other regulatory sequences that are far from the promoter. Susumu Tonegawa and colleagues discovered a regulatory sequence that was present in introns and was required for transcription to occur. These results were remarkable because they found that the regulatory sequence was far from the promoter and was present downstream rather than upstream from the promoter, within the actual sequence of the gene. Such regulatory elements that are far from the promoter are called enhancers. All eukaryotes have enhancer elements within their DNA. These could be more than '100,000 bases away from the promoter. Enhancers can be located in different places, including introns or untranscribed 5' or 3' sequences that flank the gene they regulate. There are several different types of enhancers. Many genes have more than one enhancer. Enhancers can work even if their normal orientation is flipped, meaning that they read in a different direction than the gene. Enhancers can also work even if they are moved to a new location on the same chromosome. As shown in this image, in addition to promoter proximal elements that help regulate gene expression, there are additional elements called enhancers located far away from the promoter that help regulate gene expression. So we have one enhancer in this example that is upstream of the promoter and another one that is downstream of the promoter within the actual DNA sequence for the gene in an intron. And we also have a third sequence that regulates the same gene that is present downstream of the gene. If we were to cut any of these enhancers out and change their location within the same chromosome, they would still be able to regulate the expression of this gene. Enhancers are called that because they positively regulate gene expression when they bind regulatory proteins. This leads to an increase in transcription. DNA sequences that carry out the opposite function are called silencers because they bring about a decrease in gene expression after binding regulatory proteins. The discovery of enhancers and silencers resulted in redefining the gene as the DNA that codes for a functional protein or RNA molecule and the regulatory sequences required for expression. So far, we've looked at the regulatory sequences present within DNA that either facilitate or repress gene transcription. Now, let's look at the role that regulatory proteins play in this process of regulating gene expression. Different cell types express different genes because they have different histone modifications and contain different regulatory proteins. This is what leads to a difference in a muscle cell and a kidney cell, or a heart cell and a brain cell. And the process that gives rise to these different cell types is called differential gene expression, which is a result of the production or activation of specific regulatory proteins that leads to certain proteins expressed only in certain types of cells. For example, muscle cells have the proteins myosin and actin that help in muscle contraction, whereas brain cells have more ion channels that help in transmitting nervous impulses throughout the body. Eukaryotic genes are turned on when specific regulatory proteins bind to enhancers and promoter proximal elements. They are turned off when regulatory proteins bind to silencers or when chromatin remains condensed, preventing these proteins from binding their respective sequences within the DNA. All of the regulatory sequences and regulatory proteins that we have talked about so far need to come together and assemble into the initiation complex in order for transcription to begin. Two broad classes of proteins bind to regulatory sequences at the start of transcription. The first class of proteins are called regulatory transcription factors. These bind to enhancers, silencers, and promoter proximal elements. They're responsible for the expression of particular genes in particular cell types and at particular stages of development. The second class of proteins are called basal transcription factors. These interact with the promoter, are not restricted to particular cell types, are required for transcription, and do not regulate gene expression, meaning that they are necessary just for transcription to begin, not to either increase or decrease the level of transcription. In addition to the initiation complex, another complex called the mediator needs to assemble in order for transcription to begin. Proteins that are part of the mediator complex do not bind DNA, but create a physical link between regulatory transcription factors and basal transcription factors. So here's how transcription initiates in eukaryotic cells. First, regulatory transcription factors bind to DNA and recruit the chromatin remodeling complexes, or HATs, also known as histone acetyltransferases. These enzymes then remodel chromatin, resulting in the loosening of the chromatin structure, exposing the promoter region. After this, additional regulatory transcription factors bind enhancers and promoter proximal elements. They also interact with basal transcription factors that bind the promoter. Then, all of the basal transcription factors form the basal transcription complex that recruits RNA polymerase so that transcription can begin. The construction of the basal transcription complex requires the daughter-binding protein to be bound to DNA. It depends on interactions among regulatory transcription factors that are bound to the various regulatory sequences within DNA, including enhancers, silencers, and promoter proximal elements. The construction of the basal transcription complex results in a large multimolecular machine that is positioned at the start site and is able to start transcription. This completed basal transcription complex may contain as many as 60 different proteins. This image shows a model of transcription initiation. DNA is initially tightly wrapped in its condensed form around histones. Note here that DNA is in a constant state of flux inside the nucleus, and sometimes parts of chromatin relax, allowing proteins to remodel chromatin and expose regions that aid in the start of transcription. These regions contain many of the regulatory sequences which we discussed earlier, including promoter proximal elements and enhancers. Regulatory proteins, for example activators, then bind these regulatory sequences within the DNA that then facilitates the assembly of basal transcription factors at the promoter, which then leads to the recruitment of RNA polymerase II to complete the assembly. The mediator complex proteins connect the regulatory proteins to the basal transcription complex, finally leading to the start of transcription of the gene. This concludes the section on regulation of transcription. On going through this information, I'd like you to be able to respond to the questions listed on this slide. Thank you.